Meiosis is very important for the human body to produce gametes, or sex cells such as the sperm and egg cell, that have a haploid 1n number of chromosomes. As the sperm and egg merge during fertilization to form a diploid 2n cell, once more, the process of meiosis is important to prevent a doubling of chromosomes every cycle. Meiosis also allows for changes in genetic information in order to increase diversity in the offspring. This is especially important for the survival of eukaryotic species, as a large amount of diversification ensures that a diversion from a preferred environment, whether that be a change in climate, a virus, or a new predator, won't kill all the organisms in the species. Here we have two cells about to undergo meiosis. There are two types of meiosis in the sense that oogenesis, or the creation of an egg cell, takes place in a female's body, and spermatogenesis, the creation of a sperm cell, takes place in a male's body. Meiosis is kind of similar to mitosis in the sense that the process of meiosis involves prophase, metaphase, anaphase, telophase, and then finally cytokinesis of the cell. However, there are two main stages of meiosis in which the nomenclature is to add the suffix of 1 or 2 depending on the stage. This is because cells actually divide twice in meiosis, resulting in 4 haploid or 1n cells. Both phases are important to ensure that 4 haploid or 1n gametes are produced. Here is a diploid cell with 4 pairs of chromosomes. However, before we get to a diploid cell with 4 pairs of chromosomes, the cell must undergo interphase. The preparation of a cell destined to divide meiotically is the exact same as a cell destined to divide mitotically. There will be a G1 or growth 1 phase, where the cell grows and synthesizes materials, some of which are enzymes that the cell needs to grow. In the G1 phase, there are 4 singular linear chromosomes. Before we can move on, however, there is a checkpoint ensuring that nothing has gone wrong while the cell grew. That means things like DNA damage or the size of a cell. However, when we get to the S, or synthesis phase, the genetic material of the cell is replicated. In this case, this would turn the 4 singular chromosomes from the G1 phase into 4 pairs of chromosomes, like the cell that was shown beforehand. Each of the cell's chromosomes is duplicated to become 2 identical sister chromatids. They are still attached to each other at the centromere, and it's important to note that the chromatids are still in the form of chromatin, not in a fully coiled and densely packed chromosome state. That happens later. Then the G2 phase happens, where after the S phase is completed, cells must check the duplicated chromosomes for any errors, making repairs or scrapping cells completely. After the cell goes through one final checkpoint before meiosis, during the G2 phase, the cell can start dividing. The first stage of meiosis 1, the first division of the cell, is prophase I. Here, DNA coils tightly around histone proteins to form chromosomes, becoming visible under a light microscope. The nucleolus and the nuclear membrane disassemble. Here, homologous chromosomes pair together in a process called synapsis and form pairs of chromosomes consisting of 4 total chromatids, known as tetrads. Here, something called crossing over can occur, where portions of chromatids on a homologous chromosome can break off and attach to adjacent chromatids. This is one thing that allows for genetic diversity in a species, as explained before. Then the centrosome of a cell duplicates, forming 2 daughter centrosomes which migrate to the poles of the cell. This produces spindle fibers and forms the mitotic spindle. Then, Metaphase I starts, where tetrads now line up randomly along the equator of the dividing cell, with each chromatid being roughly perpendicular to the poles of the cell. Spindle fibers from one of the poles attach to the centromere of one homologous chromosome, and then the other homologous chromosome becomes attached to the opposite pole by spindle fibers from that pole. Anaphase I follows Metaphase I, and this is where each homologous chromosome starts moving to an opposite pole of the dividing cell. This is another example of genetic variation, as the tetrads of homologous chromosomes are separated somewhat randomly, and this is called independent assortment. Finally, in Telophase I, the chromosomes have reached opposite poles of the cell, and the nuclear membrane reforms in some cells. Whether or not this happens is not important, and you'll see why next. The cells undergo cytoplasmic division through the process of Cytokinesis I, and this forms 2 new cells. Meiosis I cuts the amount of chromosomes in half. The original diploid cell had 2 copies of each chromosome, but the new cells are haploid, having only 1 copy of each chromosome. But we're not quite done yet. As said, it doesn't matter if the nuclear membrane reforms or not, because Meiosis II occurs shortly after Cytokinesis I, or the end of Meiosis I. Meiosis II is identical to Meiosis I, but it is not preceded by the copying of DNA, as that would kind of defeat the whole purpose of meiosis. The chromosomes condense, and the nuclear membrane dissolves, and spindle fibers form in Prophase II. Then the spindle fibers attach to the chromosomes, which line up at the equator of the cell, in Metaphase II. In Anaphase II, centromeres divide, sister chromatids move to opposite poles of the cell, and really the only difference here is that the sister chromatids are now separated, leaving singular chromatids. Then in Telophase II, chromosomes reach opposite poles of the cell, the nuclear membrane reforms, and then cytokinesis II occurs. This divides the original cell for the second time, yielding 4 total cells stemming from the original cell, each with half the original amount of chromosomes. It should be worth mentioning that in males, 4 functional sperm cells are produced, whereas sometimes in oogenesis, cytokinesis happens unevenly, and the result is 3 polar bodies, which are cells that are too small to be usable by the body, and 1 mature egg cell that can be used in the process of fertilization. Polar bodies often disintegrate or die out a short time after they are formed. We have now made either egg cells or sperm cells, depending on the sex of the organism. Eventually, a sperm cell and egg cell will meet in the process of fertilization, and will be the first steps to becoming a baby. This baby will have completely different genetic traits compared to any other human being on the planet. And that is the wonder of meiosis.